how to interpret trials, so save all of your questions for then, but we'd like to invite the next speaker.
And the title of the next talk is a novel ultrasensitive Huntingtin assay to measure Huntingtin protein in polyglutamine-length-independent manner. Douglas Macdonald. He's a drug hunter, molecular neuropharmacologist, has experience in drug discovery and development in industries. His lead activities include target validation, HDS, lead optimization, in vivo analysis, biomarker development and validation. The floor is yours.
Thank you very much, and good afternoon, everyone. I'd like to also thank the organizers for giving me this invitation to address you today. And I'm going to be talking about, as said, the novel ultrasensitive assay to measure Huntingtin protein in a polyglutamine-length-independent manner. This was actually just published. I'm going to talk about the data from the publication, and its also utility in a large clinical sample set. So as very poignantly expressed by Astri yesterday and also some people today, it's been a tough couple of years for our field. But one of the common things that all of these trials had was that they used our mutant Huntingtin ultrasensitive assay to detect mutant Huntingtin in CSF. And I really hope that we can look at that data carefully and hopefully learn what went on in some of these trials, the degree of lowering. In one case, they didn't observe as much lowering as they wished, so that's one of the reasons why they halted it. Another, low dose seems to be okay, high dose maybe not so much, so let's do the comparison of how much lowering we observed in those two cohorts. But despite this particular assay being used across these clinical trials, there's still a great need for additional Huntingtin protein biomarker assays for clinical use. And on the left is a very nice diagram from a review by Sarah Tabrizi et al., showing all of the different points of intervention for at least the Huntingtin lowering approaches that are out there. I actually did something at CHI where I collated all of the companies that I know of who are working on Huntingtin therapeutics, and beyond Huntingtin lowering, I came up with a list of 56 companies, and I spoke to someone today, or actually yesterday, so I'm going to change that to 57 companies. That's remarkable. Many shots on goal, and also many mechanisms of action being tested. But for the Huntingtin lowering therapeutics, these Huntingtin protein biomarker assays are really valuable because they can show you a pharmacodynamic effect of your drug as it lowers Huntingtin. At CHEI, we have a Huntingtin quantitation assay platform, and I lead that effort with a team. And we have a couple of goals. Since many of these companies who are developing Huntingtin lowering therapies are targeting both the wild type and the expanded mutant Huntingtin protein, we thought the development of an assay that would detect Huntingtin protein in a polyglutamine-length-independent manner, so by that I mean both wild type and mutant Huntingtin combined detection versus the assay that's been used, which is selected for mutant Huntingtin. And sometimes I'll call that total Huntingtin, although the reviewers didn't like me using total because there isn't really an assay out there that will detect every single molecule of Huntingtin protein in a biosample. But if I slip and I say total, what I mean is wild type and mutant combined detection. We also have as one of our goals, we'd like to develop assays with increased sensitivity, especially to be able to measure Huntingtin protein in pre-manifest HD patient CSF samples, or equivalent to the ISS stage 0 and 1. And then lastly, I'm not going to be talking about the other assay development efforts that we have ongoing, but there's a clear need for a Huntingtin exon 1 assay. We believe it really requires novel neoepitope antibodies, and we actually have a panel of those now that we're evaluating. These would be highly selective for that neoepitope on the carboxy terminus of the exon 1 protein. And as we saw earlier today, there's a wild-type Huntington assay, and we're going to be looking at that as well. We think it requires a bit further validation and refinement with regard to the MW1 pull-down step, especially because we know MW1 can recognize the wild-type Huntington, so we would like to ensure that that step is not depleting any of the wild-type. So for our polyQ-independent assay, we want it to be applied to CSF biosamples, and for that, we require an ultrasensitive assay technology. At CHDI and with our collaborators, we've developed a slew of assays, immunoassays, to detect Huntingtin proteins on the MSD platform, but that is not sensitive enough to detect Huntingtin in patient CSF samples. So we use the SMC, or single molecule counting instrument, now made by Millipore, but originally by Singulux. There's the Arena instrument in the middle there, and on the bottom right is their next generation SMC X Pro. The difference between those two is the Arena uses a fluidics methodology to detect individual samples through a confocal laser and literally counts molecules, hence its name. And the X Pro is a plate-based assay where the laser comes up from underneath the plate and takes an average of the molecules or the signals in each well. And we've actually cross-validated the same assay on both instruments, and we get identical results, so that's good. So when we wanted to go about developing this assay, polyglutamine-length-independent assay, or total assay, we started with two assays that were on our MSD platform, the 2B7 MAB2166 assay, as well as the 2B7 4C9 assay. There was a new antibody at the time that many people were beginning to use, and that's the D7F7 antibody, and so we added that into our comparative study. We also had 2B7 MW1 for mutant Huntingtin detection as a positive control for the CSF samples we were going to use in this small comparative study. I should point out the epitopes for all of these antibodies. So the 2B7 is to the extreme amino terminus of the Huntingtin protein. MW1 recognizes the polyglutamine domain. 4C9 recognizes the polyproline domain. So those three antibodies all recognize exon 1. We go outside of exon 1 for 2166, which binds about residue 450, and the D7F7 antibody recognizes residue 1220 and that domain. So when we looked at this data, only the 2B7 D7F7 SMC assay, or what we call the 143 assay, could detect Huntingtin in pre-manifest and advanced Huntingtin participant CSF samples. So we brought that assay into further validation studies. We demonstrated specificity using immunodepletion, siRNA treatment, as well as recombinant protein selectivity. We looked at the sensitivity of the assay, where we determined the lower limit of detection was 1.3 femtomolar. We looked at precision and accuracy, parallelism, dilution linearity, spike recovery. It's all in the paper that was published. I'm just going to highlight one figure from that paper, where on the left we show the sensitivity, or the LOD, being the 1.3. That's using a full-length Q46 Huntingtin protein dose response curve. In the middle, we look at the relative error and CV, and all are within the 20% variation at the concentrations that we're most interested in measuring in CSF. And on the far right, most importantly perhaps, because it's my total assay, in the red there, one of those lines is full-length Q17, and the other line is full-length Q46, and they are superimposable. So we think this assay recognizes both of those proteins equivalently. That blue line on the bottom is a large fragment of Huntingtin with a Q45, but it only goes to residue 573. And because this assay uses D7F7 that has an epitope at 1200, it lacks that epitope. So that's what we expected, something below the limit of detection. So we then wanted to test the utility of this assay, and we turned to HD Clarity, the repository of CSF and plasma biosamples. And this was the total number of participants that had donated samples, 579. And we put them through several assays, and I'm going to review 3 of the assays with you today. First, we obviously put them through our mutant Huntingtin assay, assay 40, using 2B7 and MW1, all on that SMC Arena platform. We put it through our total or polyglutamine-independent assay, the combined detection. And we also really importantly put it through the hemoglobin assay. This is a kit from Bethel Laboratories, but this is a really important point. It takes very, very little blood contamination in CSF to contribute to the signal of Huntingtin protein in your assay, because the concentration of Huntingtin in circulating blood is extremely high and the amount in CSF is extremely low. Remarkably, it can be crystal clear and still have a hemoglobin content that skews the results. So I think most people need to be looking at hemoglobin, and if it's above a certain threshold, as we published, I forget what year it was, but it was Fodale et al., that would be an unreliable data point or quantitation. So always measure hemoglobin in your CSF samples. Oh, and lastly, these 580 or so samples were run in 2 batches because they were very large, and we were running multiple assays. So I'm going to show you the combined data and presented by stage of Huntington's disease. And here, just like previously published, this is our mutant assay of looking at CSF, and you get this progression-dependent increase of mutant Huntingtin in CSF. And the next is our total assay, 143. And what you observe here is more of a equivalent level across all stages. Maybe in the advanced it's a little higher, although the N is quite low in that group. But really importantly, I want you to draw your attention to what I've circled there is we can measure Huntingtin protein in what's termed there early pre-manifest and late pre-manifest patient samples as well as controls. Remember, one of our goals was to be able to measure Huntingtin protein in that patient population where we can't with mutant Huntingtin, but we can now with this total assay. What about plasma samples that we ran? Again, with the mutant assay, you get this disease progression-dependent increase of mutant Huntingtin. And for the total assay, again, it's equivalent. All the data points are above the lower limit of detection. We then, 1 minute, we then looked at some statistical analysis of our data sets, looking at the CV as well as what I'm going to concentrate on is the interclass correlation coefficient, where it's an evaluation of the utility of the assay across a population. And the first data set I'm going to show you is mutant Huntingtin and hemoglobin assays in CSF as well as plasma on the right. Remember, we ran these in 2 batches. We still have to combine these data sets, and that will be done eventually. But what you observe immediately, and I've sort of color-coded it in green, that hemoglobin assay, 0.98, we're all set there. That's a kit. It should be good. Our mutant assay is performing wonderfully as well. 0.8, 0.8, 0.85, 0.87. That is in the good reliability, bordering excellent reliability designation. Now for our polyglutamine-length-independent assay, interestingly, The CSF data is not as strong as the plasma data. And I would argue batch 2 on the lower right, 0.979 ICC is quite good, excellent reliability. So I'm feeling that this assay may have a different utility in CSF biosamples versus plasma samples, although we still have to look more deeply into that. We need to combine these data sets, as I mentioned. There could be a biological difference. Recall, yes? I'm 5 minutes overtime? But I have 15 minutes, okay. I have one more slide. Recall the D7F7 epitope is not within exon 1, and that may be something going on in the CSF that does not occur in the plasma. Okay, my summary is that we have developed and validated a new polyglutamine-length-independent assay. It selectively measures wild type and mutant Huntingtin in a combined manner. It was shown to be ultrasensitive, 1.3 femtomolar, and reliable for the analysis of HD subject plasma and CSF samples. However, it may be more robust for plasma than CSF biosamples. Importantly, this assay provides a measure of Huntingtin in pre-manifest patient CSF samples, so ISS stage 1. And lastly, we will be looking at repeat sample and longitudinal collections in the future. I thank my contributors from CHDI, the IRBM team who both developed the assay and ran the HD Clarity samples, Doug Langbin at University of Iowa for the statistical analysis, Meta Gilling at, well, when she was within EHDN and the HD Clarity team, she helped with the logistics of the shipment of the samples, and in larger font is a big thank you to the HD Clarity study participants. And I'm sorry we may not have any time for questions, but I will be around unless you want to entertain one question.
Thank you very much. This was a very good talk. And HTT assays are particularly important. And either we move over or we have a very, very short question.
It means we don't have.
Thank you. Thanks, Doug. That was a very clear talk. So I just wondered, if you see mutant Huntington going up with disease progression, but your total Huntington assay is staying the same, how do you interpret that? Do you think wild-type Huntington's going down?
I think there's more wild-type Huntingtin in CSF than mutant Huntingtin. That's
right, okay.
And so that trend is diluted out by now adding in wild-type Huntingtin into your measure. I do.
Okay. And a quick one. I mean,
we can measure it in controls. Amazing, right?
And quickly, following on from Christian's data,
yeah.
And given that we don't know the size of a polyQ repeat in HD patient brains,
yes.
Are you going to have the opportunity to do this?
Okay, unfortunately, we have to move over.
Oh, okay.
You can ask this question later.
Saved by the bell. Yes, we'll talk, Jill. Thank you.
Okay, and the next talk is entitled an update on iMAGEmHTT.